Well, hello everyone. I am thrilled to present our image tonight on behalf of my team called Young at Heart: Progenitor T Cells Drive Immunotherapy Response. So, oh, all right, good trend. Yeah. So this image is really the product of a fantastic collaborative effort supported by funding from the Bridge Project. So this Bridge Project supports collaborations between researchers at the Koch Institute and researchers over at the Dana-Farber Harvard Cancer Center. So on the MIT side, in the JAX lab, we're interested in using genetically engineered mouse models of cancer to really understand how cancer develops with the goal of identifying new targets for cancer therapies. And we're particularly interested in understanding how to activate the immune system to target cancer cells. And we've teamed up with Sandro Santagata's lab at Harvard to apply their really cutting edge multiplexed immunofluorescence imaging platforms to take a deep look at interactions between tumor cells and the immune system through integration of molecular and spatial characterization of single cells in lung cancer. So CD8 T cells of your immune system are able to recognize and kill cancer cells, but they fail to do this in patients that present with cancer in the clinic. However, new checkpoint blockade immunotherapies are able to reactivate T cell responses. And this has led to really successful responses at controlling cancer across a subset of patients. This can, in some cases, lead to curative responses. However, unfortunately, the majority of patients still do not respond to these therapies. And so the goal of the JAX lab and Santagata lab is really to understand why this is and what we can do to extend the benefit of these therapies to more patients. So recent studies have actually identified a particular type of CD8 T cell that must be present to get a good response to checkpoint blockade immunotherapy. So these so-called progenitor cells have stem-like characteristics, and in response to immunotherapy, they can generate killer CD8 T cells that are able to control the tumor. So these progenitor cells are marked by a protein called TCF1, and TCF1 is actually being considered as a potential biomarker in the clinic to predict patient outcome in response to immunotherapy. So if you have these progenitor TCF1 positive T cells, you're more likely to respond to checkpoint blockade immunotherapy than patients that don't have these cells. However, interestingly, our recent studies in the mice have uncovered that this progenitor CD8 T cell population does not necessarily have equivalent function. So there's actually a subset of these progenitor cells is marked by another protein called CCR6 that does not give rise to killer CD8 T cells in response to immunotherapy. And hence, these cells actually don't contribute well to immunotherapy response. And it turns out that these cells are not only found in mouse, but in human as well. And this is the finding that we decided to highlight in our image in the gallery. So we performed multiplex immunofluorescence imaging, where using 3 markers, which I might be able to point to, CD8, CCR6, and TCF1, we were able to identify this subset of dysfunctional progenitor cells across most of the patient samples that we analyzed. And in further work, we were able to show in melanoma patients treated with checkpoint blockade immunotherapy that these CCR6 positive cells don't predict good response to immunotherapy in patients. And so what we hope is CCR6 could serve as an additional biomarker in the clinic in combination with TCF1 to more stringently stratify patients and to better predict which patients are going to respond to immunotherapy. And in some additional work, actually, that we're very excited about, we've also identified therapeutic vaccination as a potential approach to eliminate these CCR6 positive progenitor cells, really enriching for the functional progenitor cell population. And so we're interested in understanding how we can optimally combine therapeutic vaccination with checkpoint blockade immunotherapy to really exploit this progenitor cell population and get a greater degree of tumor control. So with that, I'll just acknowledge the 2 teams again, and I'd like to give a big shout out to Claire Rich, who actually created this gorgeous image that we have in the gallery. I'd also like to thank the core facilities at the Koch for their help with this project and their funding.